We're beginning Chapter 22, the Special Virus Cancer Program of Emerging Viruses, AIDS and Ebola, Nature, Accident, or Intentional. The next morning, figuring I had learned as much about Marburg as the experts, not including Preston, chose to reveal, I decided to look for the few congressional record citations I couldn't find in other libraries. This time, with the Davis Library basement pass in hand, I approached a tall, weighty fellow standing behind the government document librarian's desk. "Hi, I'm looking for these congressional record references," I said, pointing to a few computer printouts. "The congressional records are in the basement," he replied in a mildly inconvenienced tone. Then, with a stuffy air of library science about him, he added, "I'll have to go down to get you a few at a time. I have my own pass," I proudly displayed. "How about if you just show me where to look?" "Fine, follow me," he conceded. I negotiated the department's swinging gate, passed through a set of open doors, and skipped down a flight of stairs in hot pursuit of my guide. As I entered the huge documents room, the librarian, who had gained about 10 yards on me, pointed ahead and said, "The congressional records are over there." Then something very strange happened. Customarily, at least out of courtesy, I would have followed the man, but suddenly I felt myself entering what I can only describe as a twilight zone. It was as if time and everything else stood still. My intuition told me to turn right. Without hesitating or misstepping a step, I simply obeyed, leaving my guide behind. I had heard in uncommon intuitive leaps that led scientists to make important discoveries. I often used the example of Einstein, who explained his theory of relativity, E equals mc squared, as a breakthrough that came to him during the twilight of sleep. I suppose in retrospect this experience was like that for me, somewhat of an intuitive breakthrough, except I wasn't sleeping. Instinctively, I was being drawn in another direction, like metal to a distant magnet. I simply went with the force. Concerned my behavior, which must have seemed rude and impulsive, would further inconvenience the librarian, I blurted. "You don't mind if I have a look over here?" That, along with my abrupt departure, obviously startled him. Stumbling for words, he replied, "You're not going to find anything over there." Then, when he realized my course was set, he half obligingly advised, "That's not how you conduct research. You're likely to miss what it is you're looking for." "I appreciate that," I said, but clearly paid no heed. I just continued along my merry way down the aisle to the source of my beguilement. The librarian, forced to concede, said, "I'll get you your congressional records." We parted, him leaving me to prove myself wrong. I wandered auspiciously for about 15 yards. A voice inside said, "Turn left." I obeyed, then began to slow. My neck suddenly cranked to the left. Instantly and intently, my eyes began scanning the unmarked shelves for something I knew would be there. I just wasn't sure what it was. Momentously, my focus turned knee-high, and roamed title by title for a treasure. Then, suddenly, there it was, a faded red-covered book with white letters bleeding through. The words "Special Virus Cancer Program" touched my soul. I grabbed the title in total disbelief, completely amazed at what had just happened. All I could say was, "Wow, this is just what I'm looking for." Seconds later, the librarian walked up carrying 3 volumes of congressional records. "Here's what you wanted," he said, as he laid the text down on a desk. "I'm glad you found what you wanted." Then he turned and walked away. Still dazed as he disappeared from sight, I returned, "Thanks a lot. I really appreciate your help." For months I had read dozens of authors acknowledging the National Institute of Health's Special Virus Cancer Program. Now here was the book on it. The text covered the period from 1970 to 1971, the critical period during which I knew Gallo and Company had created numerous AIDS-like retroviruses. I thumbed through the 383-page National Cancer Institute Progress Report number 8 and realized it detailed virtually all the important investigations conducted in support of Nixon's war on cancer. It identified all the contracting agencies, summarized their grant proposals, and provided the names, addresses, and even telephone numbers of the project directors, officers, and study consultants for the entire operation. More incredibly, as I turned the pages toward the end of the book, my thumb caught on page 273. There in black and white read, Progress Report of Investigation of Carcinogens with Selected Virus Preparations in the Newborn Monkey. Submitted by Bionetics Research Laboratories, Incorporated, a division of Littleton Industries. In it was a summary of the inoculation program wherein between 1962 and 1971, a total of 2,274 primates have been inoculated on behalf of over 70 investigators in 50 different laboratories throughout the world. Here was documented evidence that Littleton Bionetics had not only been involved in the development of AIDS-like, Marburg-like, and Ebola-like viruses, but had been inoculating simian monkeys with these mutant horrors as early as 1962, and had shipped the infected monkeys dead and alive, whole and in parts, to labs around the world, including the ones in which the Marburg and Reston outbreaks occurred. Without further ado, I raced back upstairs to photocopy what on gross examination appeared to be the essential parts of the Department Health, Education, and Welfare publication. Then I did the same with its shelfmate, Progress Report 9. After about an hour of photocopying, I found a chair, sat back, and began to read the volumes from cover to cover. The task ultimately consumed the remaining part of my stay at UNC. The Special Virus Cancer Program. The Special Virus Cancer Program was introduced by the program staff of the Viral Oncology Etiology area of the National Cancer Institute. The finishing touches to the manuscript were added October 24th through 27th, 1971 at the annual Joint Working Conference. Special Virus Cancer Program at the Hershey Medical Center, Hershey, Pennsylvania, a location halfway between Bethesda and New York City, and at the same time, I noted, was fairly close to West Point, Mercktown, USA. The National Cancer Institute staff explained, the viral oncology area is responsible for planning and conducting the Institute's program of coordination research on viruses and etiological agents of cancer. Scientists within this area do not provide the broad operation management for intramural and collaborative research, but also conduct comprehensive investigations on specific animal oncogenic viruses and their interaction with host cells, and apply this information to search for viruses which may be etiologically related to the initiation and continuation of human cancer. Contract-supported research is conducted within the Viral Oncology Program under the Special Virus Cancer Program, whose primary objectives are 1. Determine whether viruses comparable to those known to induce cancer of laboratory and domestic animals are causative agents of human cancer, and 2. To develop therapeutic and preventative measures for control of human cancers when such causative agents are found. A detailed history of events leading to the development of the Special Virus Cancer Program may be found in previous annual reports of the National Cancer Institute. Briefly, in 1964, the Congress of the United States provided funds to the National Cancer Institute for an intensified program in virus leukemia research because many scientists were convinced that in an effort to identify viruses or to detect virus expressions in human tumors would contribute to the determination of the etiology of cancer. Using a new planning approach, convergence technique, an overall program aimed on the premise that one virus is an indispensable element for the induction, directly or indirectly, of at least one kind of human cancer, and that the virus or viral genome persists in the diseased individual. In 1968, the program was enlarged to encompass all types of cancer. The program plan is reviewed regularly by the director of the National Institute of Health, the director of the National Cancer Institute, the National Cancer Advisory Council, the Scientific Directorate, National Cancer Institute, and the Etiology Program Management Group of the National Cancer Institute. During the past 7 years, the Institute had developed an effective management program which has made rapid, substantial progress in understanding cancer induction by viruses. The funding level for this program in fiscal year 1971 has been about $35 million. The staff proceeded through a lengthy description of the Special Virus Cancer Program organization and management, which they summarized graphically in an organizational chart and list of managers. This followed by a list of 105 consultants of the Special Virus Cancer Program fiscal year 1971. Several doctors whose names I immediately recognized were David Baltimore, MIT Charles Chetgeny, Naval Biological Laboratories, Leon Domchowski, M.D., Anderson Hospital and Tumor Institute, Peter Duesberg, University of California, Berkeley, Sidney Farber, Children's Cancer Research Foundation, Boston, Peter Garone, Fort Detrick, Maryland, Richard Herberling and Seymour Coulter, Southwest Foundation for Research, Maurice Hilleman, Merck Institute for Therapeutic Research, John Landon, Bionetics Research Laboratories, and Brian McMahon. One of my professors at Harvard University, Albert Sabin, Weizmann Institute, Revolt, Israel, and Howard Temin, McArdle Research Laboratory, University of Wisconsin, and last but not least, Erling Jensen of Hazelton Research Laboratories. Here's the cover of what he found, the Special Virus Cancer Program Manual or booklet detailing their funding and what have you. And here was the chart that was just mentioned. If y'all want to pause it and take your time with it. I'm not reading off all this. Be my guest. It goes on for a little bit. Now, where were we? Special Virus Cancer Program highlights. Next, progress highlights of their scientific activities were presented. The majority of highlights focused on type C viruses, which of course Gallo's group manipulated by the dozens. The National Cancer Institute staff reported there are now over 100 viruses which are known to cause virtually all kinds of cancer in every major group of animals, including non-human primates. Viruses of one type C are known to cause leukemias, lymphomas, and sarcomas in chickens, mice, and cats. Particles which closely resemble type C viruses can be found in human patients affected with these same kinds of cancers. By using techniques developed for animal tumor virus studies, every effort is being made to determine whether these viruses cause human malignancies. Some important new discoveries that have been made are: 1. Certain oncogenic viruses are unable to produce malignancies unless a helper virus is present, such as Epstein-Barr viruses, thus suggesting that an interplay exists between two viruses. And 2. Certain tumor-inducing chemicals, irradiation, carcinogens, may act as cofactors in activating latent oncogenic viruses within cells. This dovetailed what Peter Duesberg had argued with regard to AIDS. And 3. Certain tumor viruses contain unique enzymes which are required in the replication of viruses in cells, exactly what Gallo and co-workers from Littleton Bionetics had been studying with the reverse transcriptase enzyme. These and other important developments will be discussed further in this report. Type C particles. When the Special Virus Cancer Program was initiated, highest priority was given to the search for human leukemia viruses resembling type C viruses causing chicken and mouse leukemias. Since that time, many type C viruses, the total is now about 85, have been found in a variety of tumors from many species of two vertebrate classes. All of these species continue to be studied intensively under the broader scope of the Special Virus Cancer Program. Several of the type C viruses are established as causative agents in leukemias, lymphomas, and sarcomas of chicken, mice, cats, and hamsters. Many of these can infect and produce malignancies in other species. Sarcoma viruses of the cat produce tumors in marmoset monkeys. Furthermore, some of these viruses can cause malignant transformation to occur in animal and human cells grown in laboratory. Cat leukemia and sarcoma viruses alter embryonic human cells. Type C virus particles have been found in association with malignancies of a spectrum of animal species, including non-human primates, rats, cattle, woolly monkey, gibbons, and man. After reading this, I reflected on Gallo's research ontology. I recalled he had not only received credit for discovering the first two types of human leukemia viruses, HTL1 and 2, but human T lymphocyte leukemia virus 1, I believe that's what it was. But he published manipulating type C virus monkey viruses. He extracted the virus's nucleic acids, infused their empty envelopes with chicken and cat leukemia sarcoma RNA, grew the mutants in human white blood cell cultures, then prompted them to infect human T lymphocytes to produce the leukemia sarcomas, general wasting, and essentially all the symptoms of AIDS, creating more AIDS-like viruses. Under the report's next heading, reactions between type C virus causing leukemias and sarcomas, solid tumors, the National Cancer Institute staff explained how and why they created more AIDS-like viruses. When inoculated into appropriate cell cultures, type C sarcoma viruses of chickens, mice, and cats produce foci, cancerous growths, of altered cells. This fundamental discovery provides a readily visible indicator reaction for the detection of sarcoma viruses. On the other hand, leukemia viruses grown in tissue culture do not cause foci or other detachable changes. The finding that leukemia viruses can either inhibit or enhance focus formation by sarcoma viruses of the same species has led to the development of methods for the detection and quantitation of leukemia viruses indirectly. Certain of the chicken, cat, and mouse sarcoma viruses are defective in that they do not produce foci in cell cultures or tumors in animals in the absence of co-infecting helper leukemia virus. Amazingly, the researchers called carcinogenic viruses defective if they were unable to produce cancers without the help of other factors including chemicals, radiation, and here leukemia viruses. Further, in the presence of a defective sarcoma virus, the helper action of leukemia viruses can be used as a specific indicator for the detection and quantitation. It is now believed that defective sarcoma virus leukemia virus interactions may be more widespread in nature than originally thought, and that similar systems may be found in man. A mouse leukemia virus, which has been adapted to grow in human cells, is now available to search for defective human sarcoma viruses, if they existed. In other words, humans would eventually be inoculated with mouse leukemia viruses in an effort to locate inactive human sarcoma viruses. Like the bovine. I gotta look that up. BLV, bovine leukemia virus. I'm guessing BLV. Yep. Like the bovine leukemia experiments Strecker described in cows were humans. Here, humans were to become the mixers for mutant leukemia and sarcoma viruses. Here, in almost lay terms, the National Cancer Institute staff described their greatest accomplishment in 1971 was to produce AIDS-like viruses for pre-cancer diagnosis. In continuing this effort, they reported an alternative approach had been developed for the detection of possible human leukemia viruses. A defective mouse sarcoma virus and its leukemia virus helper can be made to form tight functional aggregates which behave as one virus. Using a mixture of mouse sarcoma virus and cat leukemia virus, a hybrid aggregate which could be grown continuously in cat cells was produced. Again, this is exactly what Gallo reported. Because the aggregate is defective, it requires the simultaneous presence of a cat leukemia virus for producing altered foci in cat cells. Thus, a foci-forming sarcoma virus of the mouse, artificially changed to one possessing ineffectivity for cat cells, can now be used in cultures for the detection of cat leukemia viruses. The hybrid virus, as well as cat leukemia virus, will also grow in human embryonic cells and tissue culture. If sufficient amounts of the type C particles found in association with human leukemia can be obtained, the possibility exists that the cat-adapted mouse sarcoma virus can be hybridized with the human agent to produce an indicator system for the detection of human leukemia viruses. And I will show you the figure. A technique devised by National Cancer Institute researchers, including Robert Gallo, as an alternative approach for the detection of possible human leukemia viruses. A bizarre series of cancer research experiments in which numerous leukemia and sarcoma virus from cats and mice were mixed to produce a variety of mutants. One such virus infectious for cats was cultured in human embryonic cells to develop a diagnostic test for human leukemia. Take your time with it by pausing. Littleton's group in Uganda. The National Cancer Institute staff went on to explain how type C virus particles can get into the genes of normal cells and cause them to develop cancer. Then they discussed in great detail the enzyme that represented a dramatic breakthrough in the investigation of cancer. Without naming Gallo, they discussed his explicit area of expertise, RNA-dependent DNA polymerase, and explained, as Gallo had in numerous publications, the biochemical pathways of tumor virus infection and replication. Intensive investigations have now revealed polymerase activity in cells of patients with acute lymphoblastic leukemia. More preliminary evidence has shown the enzyme is in cells of sarcomas, Burkitt's lymphoma, and breast cancer. Since the RNA-dependent DNA polymerase is apparently always present in the RNA tumor viruses of animals, its discovery in the human tumor cells offer a good supportive evidence that viruses are associated with cancers in man. The RNA-dependent DNA polymerase of human leukemia cells is inhibited by a drug, rifamycin, which Gallo had received from another documented Nazi employer and biological weapons contractor, Dow Chemical Company, and which he subsequently studied and reported on, and which also inhibits the enzyme activity found in the type C RNA tumor viruses of animals. Studies are underway, principally in Gallo's lab, to explore the action of this drug and the various modifications of it. These investigations could provide new approaches to the treatment of malignancies in man. Following a shorter review of type B viruses thought to be associated with breast cancer and herpes type viruses associated with some forms of chronic leukemia, lymphoma, and postnatal carcinoma, the National Cancer Institute staff focused on Epstein-Barr virus. Their program, they said, had studied the significance of Epstein-Barr virus extracted from Burkitt's lymphoma and postnatal carcinomas. That the Interagency for Research on Cancer in the West Nile district of Uganda. This, of course, was close to where Preston noted AIDS, Marburg, Ebola, and Reston viruses were believed to have originated. The purpose of the National Cancer Institute and NARC, what's NARC mean? NARC. It doesn't have it. The purpose of the National Cancer Institute NARC collaborative effort was to determine the feasibility of further studies on Epstein-Barr virus in relation to Burkitt's tumor. Also mentioned was the fact that Epstein-Barr infection has been associated with the development of infectious mononucleosis in young adults, a disease with attributes of a self-limiting leukemia. Now it all seemed to fit. Gallo's group at Littleton Bionetics, unlike any other, had performed the core services needed during the Special Virus Cancer Program for the National Cancer Institute to achieve its principal goals. Not only had Littleton Bionetics had been contracted to manage the operations of the entire National Cancer Institute's Frederick, Maryland Cancer Research Center, including more than 750 scientists and support personnel, but here was evidence they ran the show in the West Nile district of Uganda as well. Reflecting on what I had just read about Littleton Bionetics being the world's principal supplier of monkeys during the 1960s and early 1970s. See the first 4 grant summaries in Figure 22.5. They obviously maintained a facility in this area, close to where Orbital Transport Missiles Ltd. operated. Thus I realized the unnamed animal supplier who sent Marburg-infected monkeys to Europe was undoubtedly Littleton Bionetics. Moreover, I knew Gallo had worked with Epstein-Barr virus to make his retroviruses grow faster. I was certain he knew his cell lines needed to be free of Epstein-Barr virus lest they continue to mass-produce retroviruses. But here was the most damning evidence that the entire French-American fracas was a farce, truly a distraction. This document proved that Gallo's group was intimately involved in Epstein-Barr virus studies simultaneously in Bethesda and Uganda, which would also explain how Littleton Bionetics ended up with the patent on mononucleosis blood test. Into the future with Orbital Transport and Missiles Ltd. and Gallo. Next, the report provided their projections of how their research would be developed or expanded into the future. Human studies, they wrote, would be expanded to identify viruses or detect virus expressions in human tumors and to search for candidate viruses or subviral products which induce human malignancies. Such studies were desired, 1. To identify and isolate candidate viruses or subviral products in leukemias, lymphomas, and sarcomas. 2. To identify and isolate candidate viruses or subviral products in breast, lung, and other carcinomas. 3. To develop methods for the direction of high cancer risk groups, individual susceptibility of predisposition to transformation by human viruses. 4. To develop reagents for mass diagnostic screening for candidate viruses. 5. to characterize biologically and biochemically presumptive viral agents. 6. To increase emphasis on understanding the relationship of environmental agents, chemical carcinogens, as cofactors in viral carcinogenesis. This represents a major expansion of effort requiring combined efforts of the viral oncology and chemical carcinogenesis areas. This later function was one Littleton Bionetics' chief revenue-producing specialties. The Special Virus Cancer Program's projections regarding future molecular studies, which again was Dr. Gallo's forte, and their immunological studies in which Hilleman, Krugman, and Poitze majored were summarized. Next, the Special Virus Cancer Program's summary reports reviewed progress which had been made in every working group. In this section, the staff disclosed the principal researchers directing each group an area of investigation. Under another heading, Other Research Developments in the Viral Leukemia and Lymphoma Branch, the staff described a variety of viral experiments, including those using Epstein-Barr virus, cat leukemia sarcoma, and monkey leukemia lymphoma sarcoma viruses. Here they announced a major breakthrough. Herpes simplex is a DNA-containing virus indigenous to the squirrel monkey. Recent studies have shown that this virus will induce acute lymphocytic leukemias as well as lymphomas and rectilium, cell carcinomas in owl monkeys and marmosets. This is the first demonstration that a virus produces leukemia and lymphomas in primates, and may serve as an important model for human leukemia and lymphoma. This disclosure obviously foreshadowed the announcement made almost 10 years later by Gallo that he had received human T lymphocyte virus 1 and leukemia virus he identified as the cause of T-cell leukemia in Japan. I thus wondered whether human T lymphocyte virus 1 had been developed by Gallo and company during his early 1970s. I thus wondered whether human T lymphocyte virus 1 had been developed by Gallo and company during the early 1970s as part of the Special Virus Cancer Program. Human T lymphocyte virus 1, like HIV or human immunos. I can't believe I don't know what HIV means. Human immunodeficiency virus, I was right. As part of the Special Virus Cancer Program, the human T lymphocyte virus 1, like human immunosuppressive virus, and the Marburg viruses may have been accidentally released or deployed to initiate the Japanese epidemic, for which Gallo could later claim the discovery of its etiological agent. This would put a new spin on what Schultz described as something of a backward scientific affair, I realized. In fact, the entire human T lymphocyte virus 1 saga, in light of this evidence, was reasonably a possible pilot study or model for the AIDS epidemic a decade later. Here's the figures, or what they were talking about. I'm not reading all this. Just not going to. Be my guest. One more. A cat owner's nightmare. Another paragraph dealt with cat owners and AIDS-like diseases in cats. Having studied feline leukemia sarcoma viruses, the staff reported, about 1.5 years ago, a nationwide effort was initiated to obtain cancerous cats, primarily leukemia and sarcomas, from cat breeders and owners. The National Cancer Institute put out a nationwide call for materials and cancerous purebred cats with pedigrees. Letters were sent to cat owners soliciting cats with veterinarian-diagnosed cancers. The author wrote, "The procurement effort not only obtains valuable research material, but reassures the cat owners and other concerned citizens of the National Cancer Institute's active program for the evaluation of the unlikely hazard of this disease to human health." How ironic, I thought. The National Cancer Institute was spending hundreds of millions of taxpayer dollars to prove that animals, including cats and monkeys, were potential reservoirs for cancer viruses at the same time they assured people that it was safe to handle these animals. Then, even more bizarre was the fact that as pet owners supplied the National Cancer Institute with experimental cat viruses, the researchers engineered them to cause cancers in humans. Apparently, I realized cancer virus infected animals posed less of a risk to the public's health than did the National Cancer Institute. Ain't it the truth? Filling in the missing pieces, the staff continued to describe dozens of studies in which special viruses were bioengineered in order to identify more viruses, develop a better understanding of cancers, and produce potential experimental vaccines. A list of 120 contracts given to a couple dozen special virus cancer program contractors was provided in this section. Their Table 2 included each contractor's name, telephone number, and project title. Contract summaries followed this list, Figure 22.5. Selected reports from this section, including those of Bionetic Research Laboratories Incorporated, Merck & Company Incorporated, Public Health Research Institute of the City of New York, the University of California, and Hazleton Research Labs, also spelled Hazelton in the same government document. These Special Virus Cancer Program contract summary reports filled in many of the gaps in the knowledge I had concerning the relationships between the various suspects and their activities. For instance, Gallo's boss, Robert Maneker, was commonly cited as the chief of the viral biology branch and chairman of the etiology area for the entire Special Virus Cancer Program. Dr. Roy Kinnard served as vice chairman until March 2, 1971, and Dr. Jack Grubner, who received the vast majority of contracts, carried the title of executive secretary under Maneker. Maneker and Grubner administered the Merck & Company Incorporated project entitled Research on Oncogenic and Potentially Oncogenic Viruses, Large-Scale Virus Production and Vaccine Development, directed by Maurice Hilleman. Maneker was the principal project officer on the contract as well as the segment chairman. He was also the National Cancer Institute's project officer for Hazleton Laboratories, studies on the etiology of canine cancer. Surely Preston would love to see this, I thought facetiously, as I read that Hazleton not only supplied the National Cancer Institute with viruses, but they conducted virtually the same experiments that Gallo had performed on monkeys using dogs. Their summary in Figure 22.5 showed they inoculated dogs, kittens, and mice with leukemia sarcoma viruses in a failed attempt to develop vaccines for immunotherapeutic control of naturally occurring cancers in humans. A Bionetics contract summary disclosed that Dr. John Landon served as the director of Littleton Bionetics along with David Valerio and Robert Ting. Ting had co-authored numerous Gallo research publications. The Special Virus Cancer Report noted that Gallo served as the National Cancer Institute project officer, overseeing several Bionetics contracts, and that Grubner and Kinnard shared this responsibility with Gallo. Also, see Figure 22.5, already showed it. This report confirmed that Bionetics and Merck had co-investigated African green monkeys and both challenged them with the Australia antigen associated with hepatitis B transmission. Both companies were involved in the research that led to the development of Merck's infamous hepatitis B vaccine, most plausibly responsible for spreading AIDS throughout the world. While reading the Special Virus Cancer Program research reports, many of the grimmest suspicions proved to be true. For instance, the Merck and Company contract gave for its proposed course. I'm reading that over. While reading the Special Virus Cancer Program's progress reports, many of the grimmest suspicions proved to be true. For instance, the Merck and Company contract gave for its proposed course. The investigators will devote initial efforts to developing methods for propagation, purification, concentration, and specific quantitation of candidate viruses suspected or shown to cause cancer in man. At the present time, investigations will be focused upon herpes-type DNA viruses and B and C-type RNA particles, that is, viruses known to cause lymphomas, leukemias, sarcomas, general immune suppression, and wasting, all commonly associated with AIDS. Parallel studies to evolve live attenuated and killed viruses in the appropriate animal model systems will be conducted. Particular attention will be given to developing and applying optimal methods for viral attenuation, viral inactivation, viral quantitation, vaccine safety assessment, and vaccine potency assay. Thus, in 1971, Merck's research agenda, National Institute of Health Contract 71-2059, completely overlapped Bionetics' work. Contract National Institute of Health Contract 71-2025, a highly incriminating fact which might best explain both the rapid spread of human immunosuppressive virus following Merck's vaccine trials, as well as the simultaneous AIDS autoimmune deficiency outbreaks in Central Africa and New York City. Just to make sure. It's acquired immunodeficiency syndrome. Yeah, that's right. AIDS. Acquired immunodeficiency syndrome. Additionally incriminating in this regard was another Bionetics contract that called for the company to supply support services for Special Virus Cancer Program, NIH Contract 69-2160. The Littleton Group was paid not only to supply monkeys and biochemical testing reagents to other research groups, including those at Merck, but also to compare and manipulate AIDS-like viruses in Africa and America simultaneously. Begun on June 27, 1969, and completed within a year, their work evaluated Epstein-Barr virus levels in blood taken from American patients with Burkitt's lymphoma and age and sex-matched patients with acute lymphocytic leukemia, African Burkitt syndrome, and non-malignant diseases at the same time in the same study. To complete the Bionetics contract, the National Cancer Institute of Officer Basson and his co-worker, Dr. Painta, were to rescue and isolate viruses from undifferentiated sarcomas and from human patients by co-cultivation, hybridization, and other techniques. Just as I theorized, had Orbital Transport and Missiles Ltd. Assisted in the development of testing of AIDS-like viruses, they would have received their experimental reagents and animals from Littleton Bionetics. Adjacent, this Bionetics research report was one coded National Institute of Health 71-2149. Here, $75,000 a year went to the Massachusetts Institute of Technology. That's what it means, MIT, Massachusetts Institute of Technology, for essentially identical studies on RNA-dependent DNA polymerase, on which Gallo's team had reported. The project director, David Baltimore, the researcher Streckert had accurately cited as a pioneer of the work Gallo made famous, and National Cancer Institute Officer Bionetics Overseer George Todaro performed this series of studies to identify the genetic codes in cancer viruses that enable them to cause human tumors. This work, the progress report stated, had been given the highest priority in the Special Virus Cancer Program, 22.6. Another contract Another contract of interest was the production of simian virus and homologous antisera, NIH National Institute of Health 69-93, directed by Seymour Coulter from the Southwest Foundation for Research and Education. See final contract, figure 22.5. Here's 22.5. Here, the summary revealed Coulter's group served as quality control experts for the National Cancer Institute's chief monkey virus suppliers, principally Littleton Bionetics. In addition, Coulter was responsible for diagnosing the viral isolates that may emerge from studies done. In other words, as new forms of mutant monkey viruses emerged from Bionetics and other labs, Coulter diagnosed what kind of bugs they were and what they might be expected to do in monkeys and man. I suddenly realized where the phrase emerging viruses came from. In summary, I realized this special virus cancer program report documented 3 frightening facts. 1. The Littleton and Merck network of researchers undoubtedly produced an array of AIDS-like viruses under these contracts alone. 2. Such work was going on at the time Merck hepatitis B vaccine studies were being conducted in New York and Africa under Krugman and Hilleman. These men shared their information and resources. And most alarming, 3. Robert Gallo's affiliates at Littleton Bionetics supplied the experimental viruses and monkeys, both infected and uninfected, to National Cancer Institute and Merck researchers in Bethesda, Central Africa, New York, and Pennsylvania. Therefore, had either one infected monkey or a bit of a monkey virus contaminated material been used to develop one batch of experimental hepatitis B vaccine in any of their labs, this alone might explain the origin of AIDS and the epidemic's simultaneous emergence in New York and Central Africa during the 1970s. Here's, what is it? This is 22.5. There's 22. Pause and read. Pause, zoom and read. I don't care. I'm not reading these. Yeah, this goes on for too long. That's it.